Dear students, our today's topic is gene mapping or recombination mapping with 3-point test cross. And in this type of test cross, we try to measure the relative distance between genes located on same chromosome, and those genes are known as linked genes, and how far these genes are situated with respect to each other. And then this is the method, indirect method to identify their respected distance with each other. In this test, the recombination frequency is tested when more than 2 genes are taken, and the map distance, or simply the distance between 3 genes located on same chromosomes, are used. That we try to measure the distance between 3 genes, and this experiment was first carried out by C.B. Bridges and T.M. Albrecht on Drosophila. And as the wild type Drosophila male having one X and one Y chromosome and has 3 genes attached on X chromosomes. These 3 genes are linked genes, means these 3 genes are present on X chromosome, and these are represented with SC plus, EC plus and CB plus. This plus indicates that these 3 genes are normal copy, and these wild type Drosophila male are crossed with females having 2 X chromosome, and these females are homozygous for all the 3 mutant genes. It means these 3 genes, those are linked genes or present on X chromosome, are mutated copy or present in mutated copy on both X chromosome of the female Drosophila, and then a cross is made between wild type male Drosophila having 1 X chromosome with this genetic combination, that all the 3 genes are in normal copy, and with the cross is made with female Drosophila having both the copies in mutated form present on 2 X chromosome. So when this type of cross is made, the F1 females carries 1 normal copy of X chromosome and having normal copy of all the 3 genes descended from the male parent. Definitely the X chromosome is transported from the male parent, and that X chromosome is in normal copy, that all the 3 genes present on X chromosome are in normal version or in normal copy, that these 3 genes are producing their normal phenotype, and another X chromosome is descended from mutated female parent. So the F1 females are heterozygous. It means 1 X chromosome is having all the 3 genes in normal copy, and the other X chromosome having all the 3 genes in a mutated copy. And as this is a recessive mutation, it means that the normal copy will express its phenotype, and the mutated copy will express only when both the X chromosome has the same mutated copy of all the 3 genes. The F1 males carried the X chromosome having mutated copy of all the 3 genes, and a test cross is a cross when F1 Drosophila or F1 plants, or here we are taking the example of Drosophila, are crossed with recessive parent. Why this is known as test cross? Because that what type of gametes are produced by F1 offsprings or F1 generation, it is ascertained, it is identified through a test cross. And in this case, the F1 females, those are heterozygous for the 3 genes on X chromosome, are crossed with F1 recessive male parent, and it is equivalent to a test cross, because in test cross the F1 individuals are crossed with the recessive parent, but here in this case the F1 males are equivalent to the recessive parent, and then this is known as a test cross. So as we came to know that all the 3 genes are present on X chromosome, and we don't know what is the correct arrangement of these 3 genes, that what is their sequence of arrangement on X chromosome, and here the following three possibilities are there. The possibility first, second, third. The possibility first says that the gene SC and CB are on both extreme of X chromosome, and the gene EC is present in between SC and CB. Similarly, the arrangement 2 and 3 is there. So our main target is to identify the correct sequence of these three genes, and this is ascertained through an indirect method, that is through crossing, and how the recombinant progenies are produced and how the recombination takes place between these three genes, and this will come. So as represented through this photograph, that these are the P1 wild type male, and this is mutated female, and this mutated female has all the three genes in mutated copy present on both the X chromosome, and their mutation is represented with skewed bristles and echinus eyes and cross-veinless wings. These three genes are for three mutated phenotype, and as this is a recessive mutation, it means one normal copy of each gene will express the normal phenotype or wild type phenotype. So a normal wild male is crossed with a mutated female Drosophila, and then the F1 progeny is obtained, and this, as this is a test cross, so F1 progeny is crossed with a recessive parent. So in this case the F1 females are the heterozygous Drosophila female, and these heterozygous Drosophila female shows the normal wild type phenotype because one normal copy of each gene is present, and these F1 females are crossed with the F1 males because each F1 male receive one mutated copy of each gene present on X chromosome, and as each male has only one X chromosome and the three genes are in mutated version, so it is equivalent to a test cross, and through their cross all class 1 to 8 phenotypes are obtained, and what type of characters these progeny shows is represented through here phenotypes, and their genotype is represented here, and the number observed, that in what number these progenies are obtained, and the total progeny is 3248. So as we know that these are two chromosomes of female Drosophila, so the crossing over takes place between SC and EC and between EC and CB, and so when crossing over do not take place, then both the X chromosomes separated to two different gametes, and then a different type of phenotype is obtained. But whenever the crossing over takes place between these genes, either between SC and EC or between EC and CE, then new or recombinant progenies are obtained, and these 1 to 8 are some are the non-crossovers or the parental combination, whereas the others are the crossovers or the recombinants. So in this case the 1 and 2 phenotypic class 1 and 2 are the parental combination, whereas the class 3 to 8 are the recombinants, and as we can see that the parental combinations are greater in number as compared to the recombinants. It means these three genes are linked and present on same chromosome, and they inhibit or anyhow minimize the recombination, because whenever the genes are closely situated on same chromosome, they interfere or they hinder or they reduces the chance of recombination, and whenever the genes are separated on a chromosome or present on two different chromosomes, there is a more possibility of recombination. So in this case, when recombination between any of the gene, that is between gene SC and EC and between gene EC and CE does not take place, then non-recombinant chromosomes are produced and the progeny shows parental phenotypes in following numbers. So as this figure shows that these genes are, suppose these genes are arranged in this way, SC, EC and CB, but still we don't know that what is the correct arrangement of these genes. It will be ascertained or it will be find out through the frequency of recombinations and their distance between these genes, that how far these genes are situated with respect to each other. But here this arrangement is assumed, and so if no crossing over takes place between gene SC and EC and between EC and CB, it means both the X chromosome of the female Drosophila will undergo for separate different gametes. It means without recombination, and will produce the parental phenotype. And how these parental phenotypes are produced? So here this is represented by class 1 and 2, and here the numbers, phenotype numbers are observed as represented in this figure, and these numbers are quite high if compared with other phenotypic classes. Now when recombination between gene SC and EC takes place, then recombinant chromosomes are produced and the recombinant phenotypes in following numbers are represented through phenotypic class 3 and 4 and 7 and 8. So when the recombination between gene SC and EC takes place, that only at one place, then new recombinant chromosomes are produced and phenotypic class 3 and 4, 7 and 8 are presented. And so how the recombination between gene SC and EC takes place and how the new recombinant chromosomes are produced, it will come in next slide. So this slide shows that recombination between gene SC and EC, that new recombinant chromosomes are produced in such a way that we can see that here this figure shows that how the genes are dislocated from one chromosome to another chromosome, another homologous chromosome, and then new combinations of genes are produced on two chromosomes. And here the phenotypic frequency or phenotypic class 3 and 4 are presented, such as that when SC moves from one chromosome, X chromosome, with EC plus and CB plus, so here SC, EC plus, CB, and similarly SC plus with EC and CB, SC plus, EC and CB, and their phenotypic numbers. And another possibility of crossing over between SC and EC is that when SC breaks from here and then take with EC plus of another chromosome and again with CV gene of the first chromosome, and another chromosome will have the same opportunity with SC plus, EC and CB. So we can see that SC, then EC plus of second chromosome and CB of first chromosome, and similarly for the lowercase SC and EC and CB. So here the crossing over further between SC and EC takes place, as well as between EC and CB. So here the phenotypic class 3 and 4, 7 and 8 are represented with their corresponding numbers. So this slide shows that when the recombination between gene SC and EC located on same chromosome takes place, the frequency or phenotypic class 3 and 4 and 7 and 8 are represented with their respective numbers 163, 130 and 1 and 1. So if we add 163 plus 130 plus 1 plus 1, it means these are new recombinants. So 163 plus 130 plus 1 plus 1 and divided by sum total of all frequency or all phenotypic classes, that is 3248. So it comes 0.091 Morgan, and as 100 Morgan, 1 Morgan is 100 centimorgan. 1 Morgan is 100 centimorgan, so it equals to 9.1 centimorgan. This centimorgan is a scale of map distance or gene distance, or the distance between genes present on chromosome, and it is in the honor of the famous scientist T.H. Morgan. Now this is the second possibility of recombination between two other genes, EC and CB. So when the recombination between these two genes takes place, then new recombinant chromosomes are produced, and the phenotypic class 5 and 6 and 7 and 8 are produced with their respective numbers as represented here. And how this recombination takes place between these two genes is represented with next slide. So as we can see that the first opportunity was that when the recombination between gene SC and EC took place, but now here the case is that recombination between gene EC and CB takes place, and here this is represented with this black lining, that how these part of chromosomes are get detached and attach another homologous chromosomes. Similarly with these red lines, it shows that how this part of chromosomes detach and attach with the homologous part of chromosome. And so here if we take that SC EC with CB plus, that is SC EC CB plus, and the number is phenotypic number, or the total individuals of this phenotype are 192. And similarly SC plus EC plus with CB, that is SC plus EC plus CB is 148. And another possibility between EC and CB recombination is that when SC attach with EC and again with CB. So in this case again this EC and CB get detached or recombine with new chromosomes, and similarly SC plus with EC and CB plus. So here SC EC plus CB was this case SC EC plus CB, and another case is SC plus, that is SC plus EC, that is EC and CB plus, and their respective number is represented with right hand corner. So this figure shows that phenotypic class 5 and 6 and 7 and 8 takes place when the recombination between gene EC and CB takes place, and their number is represented with at right hand corner. And so if we add all these new recombinants when the crossing over between or recombination between gene EC and CB takes place, then we can add that 192 plus 148 plus 1 plus 1 divide by sum total of all frequency or all phenotypic classes, and it comes to 0.105 Morgan or 10.5 centimorgan. It means the gene distance or map distance between gene EC and CB is 10.5 centimorgan. So from this slide we can definitely have an idea, or now a clear understanding should be there, that if these genes are arranged on same chromosome, that is SC, EC and CB, and their respective distance, that when the recombination between gene SC and EC takes place, then we find that the both the genes are arranged at a map distance of 9.1 centimorgan, and when the recombination between gene EC and CB takes place, then these genes shows that they are situated at a 10.5 centimorgan apart from each other. So if we represent or if we arrange all these three genes, it means these map units are additive in nature, so we can add both the distance and to calculate the total distance from gene SC at one corner of the chromosome with the gene CB arranged at or present at another corner of the chromosome. This comes total that 9.1 plus 10.5 centimorgan, and it is sum total, it comes to 19.6 centimorgan. It means genes SC and CB are arranged at a map distance of 19.6 centimorgan on a X chromosome of Drosophila. So out of the three possible arrangement of these genes with respect to each other, the first possibility, the first arrangement is found correct, and it is represented that all these three genes SC, EC and CB are arranged or are present in this way, represented with this number first. Thank you.